Hi, everybody. Thanks so much for joining today for this CABA webinar on chemicals in chalk streams. My name's Emily McCauley. I'm the Communications Assistant for Media Events here at the Rivers Trust, and I'll shortly be handing you over to Alison Matthews, our Chalk Stream Restoration Programme Manager. But firstly, I'll just go through a few housekeeping points. So captions can be enabled and can be done so by clicking the CC button at the bottom of your screen. There will be a Q&A after today's session, so please get your questions in. This can be done through the Q&A function, again at the bottom of your screen. And finally, this webinar is being recorded, but as an audience member, you won't be seen on camera, and this recording will be posted on the CABA website afterwards. So that's all from me. I'll now pass over to Alison Matthews. Thanks.
Morning. Great to see lots of you joining us here. This is our fourth CABA Chalk Streams webinar. You can get hold of the recordings for the previous events on our website, and Emily will probably post a link in the chat. So chemicals are such a topical issue at the moment. I'm really pleased to have 5 presenters today. This is really pushing the boat out for us. So we're going to start off with Rob Collins from the Rivers Trust, giving us a general introduction. Then we're going to move on to Gary Fones at Portsmouth Uni, who's going to talk us through some of the research they've been doing there. And then we're going to hear about groundwater, which often gets forgotten in the world of chalk streams and never should be. So Polly Wallace from the Environment Agency is going to talk to us about chemicals flowing through the groundwater system into our rivers. And then we're going to start thinking a little bit about how chemicals can affect the things living in the river. So Sam Green from Wildfish is going to talk to us about his work. And then we're going to wrap up with Kate Heppel telling us about citizen scientists learning more about chemicals and how the team that she works with in the Chilterns are working with their local community to try and minimise chemicals getting into the environment. So it's a really packed programme. The presentations are going to be quite short, so we're going to keep things moving along. We're going to hand over seamlessly one to another, and then we're going to have time for Q&A at the end. So please do put your questions into the Q&A, and also give us any feedback as well. If you've got feedback about the presentations or the webinar format or anything, happy to hear it, and look forward to these fabulous presentations. So I'm going to hand over as the first presentation to Rob Collins.
Thanks very much, Alison. I'll just share my screen. Give me a sec. Can someone just confirm you can hear me, but also see my screen okay?
Yep, all good, Rob.
Yeah, thank you very much. I'm feeling slightly embarrassed. You've got my blurred attic in the background rather than a beautiful chalk stream that Alison showed, but there we are. But thanks very much, Alison. Yeah, and welcome to everyone. I'm Rob Collins. I work for the Rivers Trust. I also chair the Catchment-Based Approach National Support Group. And I've been involved in chemicals work for a few years now, chemical pollution of fresh waters, and this presentation really is just setting the scene, providing some context, really a bit of background. I'm not particularly going to focus on chalk streams, others will do that, but I'll bring across some broader issues. One of the key points I want to kind of get across is that I think chemical pollution of the wider environment, including fresh water, is really a societal challenge, actually. We use chemicals right the way across society, and we need to find solutions right the way across society. So what do we know? Well, chemicals are ubiquitous in our aquatic environments, and probably most people on this call are aware of the many different sources, but also key pathways by which they are transported and emitted and discharged to fresh and coastal waters. Industrial chemicals and domestic chemicals will find their way down to the sewer system and into wastewater treatment plants, where they'll only be partially treated and thereafter discharged into receiving water or captured in sewage sludge. Come on to that later on. Obviously, that system is linked into storm overflows. That represents another key pathway at times. Road runoff with hydrocarbons and metals particularly being washed off impervious surfaces. And obviously agricultural land, pesticides, but also veterinary drugs. And that in combination means that, we find chemicals in our aquatic system wherever we pretty much look and for whichever ones we care to look for. And the upshot is that not a single, under the Water Framework Directive, which we still adhere to, not a single English river is in good chemical status. So that would include all chalk streams, obviously. Um, but that classification is based on just around 45 or so chemicals that are regulated and come with an environmental quality standard, so a concentration which should not be exceeded in order to protect aquatic life. And many of those 45 actually were banned or phased out some years ago. But we know that we find hundreds more um, other chemicals, so pesticides, metals, industrial chemicals, hydrocarbons, personal care products, pharmaceuticals, huge, great long list. We've tended to call them emerging pollutants, but I think we kind of know quite a bit about them now and their presence, and we detect them at high. We have very high detection rates for nearly all of them, actually, I'd say. But what that means, what their presence means, is not always necessarily well understood. But you can hone in on particular chemical groups, and we've done that over the last few years. So a piece of work we did looking at PFAS, forever chemicals in English rivers. That's the map on the left there. This is looking at Environment Agency data, and you can see that both the EQS for water, the water columns, so PFAS in water, but PFAS in biota in fish are pretty much exceeded everywhere. The EQS in biota is exceeded something like 200 times on average. So it's, you know, PFAS is a significant problem, but um, you can look at chemical mixtures, and we know that where you get combinations of these chemicals, you can get synergistic or additive effects. And so the map in the middle shows some output of a piece of work we did looking at, I think it was PFAS, pesticides, and ibuprofen, which the literature tells us in combination are not good for aquatic life. And the map tells a story, you find them everywhere. And just to not to forget chalk streams, the Kennet's on the top right there. It's, I think, actually the details a bit too small, I think, but again, you find tens of chemicals in both surface water and groundwater, as Alison says, not to forget the groundwater in the Kennet as well. So chalk streams are no exception to this chemical pollution. And another example here, work we did with York University over a couple of years ago, looking at pharmaceuticals in uh, national park rivers in England. So with respect to chalk streams, Norfolk Broads and South Downs will be very relevant here. And what we found was that you find a whole range of different pharmaceuticals at times at levels that will impact upon aquatic life, exceeding where they're known, exceeding environmental quality standards, and also that at certain locations and certain times you find concentrations of antibiotics that are high enough to trigger microbial resistance, and that raises implications for public health. You know, people swim in national park rivers, so what does that mean if you swallow a mouthful or two of um, bacteria resistant to antibiotics? And the other point I wanted to highlight here, and I think it's really applicable to chalk streams, is that during this piece of work, by chance, we happened to sample during the summer of 2022, as you remember, was pretty dry. It was drought in certain localities, and we found real spikes of pharmaceuticals. And I'm sure if we looked for other chemicals, that would have been true of them as well. Real spikes of chemicals coming through because through the treatment plant, you know, you get the daily discharge, and that's exacerbated by tourist numbers coming into parks. And during this prolonged drought, you haven't got the dilution, the river flow isn't there. So you get these real spikes of chemicals, and aquatic life cares about the spikes. It doesn't much care about annual averages. And so this is a real challenge, given that, you know, we're going to face more severe and frequent droughts over the coming years. And what does this all mean for aquatic life? Well, there's a huge range of lab and field studies that have shown a number of acute and chronic impacts of chemicals upon aquatic life. Probably the most well known, at least in the media like to pick up on this one, is intersex, whereby male fish take on female characteristics due to the presence of endocrine disrupting chemicals. And there's a piece of work done, I think it's, yeah, University of Exeter just a few years ago, showing that roach, so a very common coarse fish, 25% of roach exhibit this phenomenon. So it's, you know, quite a thing. And many other studies showing, and the screenshot on the right showing, for example, impacts of pharmaceuticals on fish behavior and so on, but many impacts besides. But there are also implications for human health of this chemical pollution. So I mentioned the freshwater recreation and swimmers in rivers and the chemicals we found, but we've looked recently at raw drinking water sources. So this is agency data from this map, and just simply totted up the number of different chemicals you can find in what are supposed to be drinking water, surface drinking water in this case, safeguard zones, and you can find up to 300, in fact, over 300 in some locations. Unfortunately, a lot of the chalk streams there look to have no data, but it's almost certain you'll find tens of chemicals in these safeguard zones. And despite we do have good water treatment in this country, but despite that, we know trace levels of these chemicals find their way through to our taps, our tap water. And another issue I mentioned in the wastewater treatment plant, if a lot of chemicals will get held up in sewage sludge. I think 85% of that in the UK goes onto agricultural land. So you've got issues around the soil microbes, the terrestrial ecosystem contamination. You've got issues of crop contamination. You've got issues of those chemicals running off straight back into the river. So a real issue here. So we've got some real challenges, and the question is, what do we do about all of this? How do we address the challenge of chemical pollution in our aquatic environments? And as I mentioned, I think it's a societal issue, and we need to sort of capture that in our responses. I think we can't get around the fact that you have to control chemicals at source. That's once they're released into the environment, it's so much harder to do something about them. So controlling its source is really critical. So banning the most hazardous, using them much more sustainably. So we've seen our successive governments creep very slowly towards finally banning neonicotinoid pesticides that obviously have such an impact upon polluters, pollinators, I should say, but also upon aquatic life as well. But linked to that, we've found that an important element of that control at source relates to our authorization and regulation of chemicals. And in the UK, we've really diverged from the EU REACH, which is a body that does the authorization and regulation in the European Union. We've really diverged from that over the last few years after following Brexit. So in that time, EU REACH has banned, well, this slide says 36 pesticides. I think it's up to 40 odd now, many of them very hazardous. We haven't responded in the UK. The EU has a restriction proposal on PFAS, and ahead of that, both Denmark and France have proposed bans on certain uses of PFAS. Again, I think our government is thinking about some of this stuff. We've got no hard evidence of any steps forward. And we've had the recent update to the Urban Wastewater Treatment Directive that's, well, it's got lots to it, but as in talking about improved monitoring of emerging contaminants and the polluter pays element with pharmaceutical and cosmetic industries to pay for the water treatment. So a really important development. And again, we've not yet taken that step. We absolutely need improved monitoring. I think any regulatory monitoring is always going to be resource constrained. It's always going to look at sort of spot samples of chemicals on at best a monthly basis, probably more like every 2 or 3 months, or even less than that. So that monitoring will miss high flow events, which we know, certain chemicals and certain sources will be triggering during high flow events. There's very little focus on chemical contamination of sediments and bed sediments and rivers, for example, even though we know some like to preferentially prefer to attach to sediment. We've got that issue I mentioned before, that the legislation currently only sets an environmental quality stand for around 45 chemicals, yet we find hundreds. So lots of limitations around monitoring that we need to address. I think Gary in the next presentation will touch on some of this. We do need to see greater investment into wastewater treatment. That pharmaceutical national park study I mentioned, we found a number of sort of very small rural treatment works that provide secondary treatment and can't really cope with the influx of tourist numbers. Just to add, though, that bearing in mind that a lot of chemicals can end up in sewage sludge at the treatment plant. But nature-based solutions can play a role here. The photograph on the right shows a wetland system and a treatment plant in East Anglia, and we know that these sorts of systems can attenuate chemical pollutants quite well, actually, in some cases very well. And just finally, coming back to that point around raising public awareness and this being a societal issue, we have to do so much more about making the public aware of the impact of using chemicals in our homes and gardens, particularly. You know, a lot of us have got sheds that look like this. Helping people understand where those chemicals go once you flush them down, and you know, putting paints and oils down the drain is not a good thing. And making people aware that actually there's some much less toxic products available now. I wouldn't like to say toxic free, but several, you know, cleaning products in the kitchen. Certainly there's some good products available now. And also very few people are aware of medicine take back schemes that are available, that you can take, or you should take, unopened, unused and out of date medicines and take them back to your local pharmacy. Don't flush them down the toilet or the drain, you know, particularly antibiotics. So there's so much more we can do around this particular issue. And I'll end there just to say that in my role in the Rivers Trust, but also as CABA and also within Wildlife and Countryside Link, we do undertake quite a lot of advocacy work around chemicals, and occasionally have the ear of a minister or two. And it's really important that we, you know, we come armed with as much data as possible. And we're aware of lots of really great work going on around the country, some of which you're about to hear about. But you know, if you think you've got some good data and we can use that as to add power to advocacy, then please get in contact. And I'll stop there, stop sharing, and I shall pass on to Professor Gary Phones. All yours, Gary.
Thanks, Rob. Let's see if we can do a seamless handover. Hopefully. Can everyone see?
Yeah, all good, Gary.
Okay, brilliant. Thank you. Yeah, thanks, Rob. Just a quick apologies. One, it's a good job it's online because I've got a cold, and also the university network seems to be playing up today. So we'll see. Hopefully I won't keep disappearing. Following on from Rob's talk, so what I'm going to talk about is how we go about monitoring the emerging contaminants, particularly in chalk streams. There we go. So I'll just introduce myself. So I'm Professor Gary Phones, University of Portsmouth, Fellow of the Royal Society of Chemistry. My main work is looking at water quality, sediment quality monitoring using a different type of approach to monitoring of contaminants in the aquatic environment. Currently working with a number of UK water supply companies, also with the Environment Agency, a number of NGOs, about using this passive sampler called ChemCatcher, which I'll explain this morning. And just to highlight, ChemCatcher was developed at the University of Portsmouth with my colleagues who are both now retired, so that's Graham and Richard. In terms of emerging contaminants and the work that we do with the Royal Society of Chemistry, just wanted to highlight some work that we've been doing, and from a position statement paper that came out that highlighted some different monitoring strategies, and that we have a number of meetings. So we've had one per year. We had emerging contaminants, and then one of the contaminants, PFAS, that we had this year. And so if you're interested in any RSC side of things, there's always meetings relating to emerging contaminants. So pesticides, pharmaceuticals, PFAS, all the things that Rob mentioned in his talk. So monitoring chemical pollutants. we know that we need to monitor, but how do we actually do it? So when do we collect our sample? As Rob alluded to in his talk, it's normally collecting a sample of water. However, that's not really the best approach. So there's lots of issues in terms of relating that to the real environment. And we've highlighted that there's a number of disadvantages to just collecting spot samples. So that's the cost of having to go out there all the time to collect the samples. The key one is it only provides that snapshot. So you potentially miss any other inputs coming in because you go and collect your sample, and then the following day something happens and there's an input of your contaminant which you don't pick up. Means that it's not representative of any of the conditions. So if things are fluctuating, like stochastic inputs, Rob mentioned in terms of detection limits, environmental quality standards, analytical techniques are getting better, but is there another way? Now, one of the key things here is if we're only collecting spot samples, does this lead to misleading information that we can then base management on? So some work that we did on the itching a number of years ago just highlights why spot samples might not be that useful. So this is some work, as I said, using that we looked at phosphate. We've got some EA data. We've got our data, which was collected daily. They seem to match up. However, the monthly sampling that the EA did is missing all of those large spikes, which you might think, well, is that important? If it comes to management decisions, probably yes. So if we look at the mean EA data, 0.034, which puts for chalk streams, puts it somewhere in good water quality. If we look at R1, it's nearly double, which means that's nearly into the moderate. So we're underestimating by only taking spot samples. So is there another way rather than having to go and collect hundreds of spot samples? So at Portsmouth, we developed this passive sampler called the Chem Catcher, which can be used for a number of different contaminants. It all relies on the solid phase extraction disc. We use one called an HLB, which sequesters pharmaceuticals, pesticides. We're developing one currently for PFAS. And so the system goes into the river, it accumulates over time, normally somewhere between 1 to 3 weeks. The example here is one that's over 2 weeks. And then we start to get a thing called a time-weighted average concentration. So we do miss peaks and troughs, but what we get is something that's more applicable over that longer period of time. So we can start to relate that to the health of the river. Just highlight some work we did a number of years ago with Southwest Water before we get onto our local chalk streams. Key take-home message here is by using the Chem Catcher rather than spot samples, we showed that we can detect these acidic herbicides like MCPA, Micaprop coming into the system, which the spot samples were missing. So onto chalk streams. We've been studying the River Test and the River Itchen over a number of years. The first part of the work that we started doing was looking at spot samples, even though I said that's not the best way, but this was to test our HLB receiving phase to see could we sequester a whole suite of these pharmaceuticals and pesticides in the River Test and River Itchen. So this was published a number of years ago on the Test and Itchen. We collected this, well, the spot samples were collected. It was collaboration with Southern Water. And the key caveat, there was no concentrations. It was just looking at presence and absence. The key take-home message were that there was lots of pharmaceuticals, lots of pesticides detected across a whole number of sites on the Test and Itchen. Key ones that we detected over and over again, and ones which were environmentally important. So the next step was to then change our analytical technique. So we then started to look at a targeted technique. So all of our work linked with ChemCatcher uses LCMS, so liquid chromatography mass spectrometry, to try and elucidate all of our pharmaceuticals, pesticides that we see, and then try to get some concentrations, and then we can start to see what kind of impact they're having. So one of the things that we did was we had a number of sites and the test and itching where we deploy the ChemCatcher 2-week intervals, and then we started to then get the concentrations for these so we can then compare those and try and think, how are they impacting the aquatic ecosystem in our chalk streams? So the slide you can see there is from the paper. We're trying to link that to impacts to macroinvertebrates, looking also from point source inputs from wastewater treatment plants. Key findings were we managed, with our technique, we managed to quantify 121 different compounds. This map here shows that in the headwaters of the test and itching, there's some kind of legacy herbicides that we've detected, which come through, we think, well, hypothesized are coming through groundwater. And then it then starts to change into mainly pharmaceuticals once we get downstream, and then we've got lots of point sources from our wastewater treatment. Now we've got concentrations, we found there were a number of compounds which were at or above the concentration for impacts on aquatic organisms. One of the ones to highlight is imidacloprid, which could potentially have an impact on macroinvertebrate communities. So this is from the Spot On which is the flea, the pet parasiticide treatment for your dogs and cats and any other animals. So some of the things that we've been looking at, and I won't go through all of these, but these are kind of the things that we're detecting, linking this to ecotoxicology and in terms of PNEC, but some of the ones we get for the plant protection products that we find are these things like atrazine and their metabolites, diaron, diazinon. These are all kind of legacy contaminants that which have been banned in the UK, and then we're hypothesizing are actually coming from groundwater inputs, which Polly will hopefully talk about in the next one in terms of groundwater contamination in the test and itching. We also find a whole load of pharmaceuticals and various ones which are above PNEC, so like diclofenac, venlafaxine, which then we have to take into account, is the PNEC correct? So some PNECs concentrations are relatively high. Some of them appear in the EU watch list, and then they've dramatically reduced the PNEC concentration. So in terms of the way our approach for monitoring in chalk streams, we've now got a number of projects which are still underway. So we've moved to the Hampshire Avon, where we're working with the Environment Agency and Wessex Water, looking at point source inputs from wastewater treatment plants and a number of other, and also from road runoff. So we're looking at the A303. Interesting to go from the river test to the Hampshire Avon. It's slightly different. In the winter, we see lots of floodings, which makes field work quite interesting. We're back on the river test with a NERC funded project, trying to take utilize the ChemCatcher in a number of different ways. So we're looking at point sources downstream. So we're working on the test at Fullerton, where they claim there's no spills from the CSO, which is linked to the wastewater effluent pipe, but apparently there is. We're developing a slightly different system where we can get different timings. And this is our team, which are out on the test as we speak now, deploying a new device to be able to get different timings of the chem catcher release, so we can try and look at different time frames rather than just those 2 weeks deployments. But overall, at the moment, our work is basically focusing on monitoring these contaminants of emerging concern, linking them into their impacts on aquatic life. So I'll just wrap up there and then just say thanks to all of the previous PhD students on our journey, when we've gone from looking at one particular pesticide and through to a whole suite of pesticides, now into pesticides, pharmaceuticals. We're now going to be working on PFAS, and then linking in all of this in terms of different deployment techniques of our chem catcher. We've got a new PhD student of vet meds, and then we've also had a number of undergraduates that have been focusing more on taking data and looking at macroinvertebrate or impacts on macroinvertebrates, which Sam is going to talk about a little bit later, and then some of the people that we've worked with. So I hope that's given you an idea of a slightly different way of monitoring emerging contaminants rather than just taking spot samples. So now I'm going to hand over to Polly, who is going to talk about groundwater.
Hello, I'll just share my screen. Okay, can you see that?
Can't see anything, Polly.
You can't. All right, okay.
Can see that now.
Thank you. Get there in the end. Yeah, good morning. Thanks for inviting me to talk about and raise the profile of groundwater. So yeah, I'm a hydrogeologist. I work in the Solent and South Downs area of the Environment Agency. A lot of my work involves just ensuring that our numerical models that we develop, both water resources and water quality, and also others develop, are just based on the best evidence and the best conceptual understanding. So I realize there's a lot of experts out there. This is a quick 10-minute run through, so it's directed at a wide audience. And I'm here just to talk about, yeah, the groundwater chemistry of the chalk and some of the sort of pathways in the chalk. One moment. There we go. So it's just a map to remind us where we are. It's a geology map, and it's lots of green, which shows us it's lots of chalk. I'm going to talk about some nitrate trends, both in the River Itchen and also in a groundwater monitoring point very close to the Itchen. I'm going to think about how chemicals move through our chalk aquifer. So we'll just have to have a quick think about the hydrogeology of the chalk. And also, I'm going to talk about the nitrate time bomb, the fact that we're still feeling the impacts of legacy agricultural practices, especially in the 1980s and 90s. And hopefully a bit of a good news story about atrazine. I'll let you be the judge of that. So this graph shows increasing concentrations of nitrate, both in the River Itchen at Easton and also in a groundwater monitoring point close by. So 3 points really from this graph to take away is that we're seeing very high nitrate concentrations now, higher than we've seen. The fact that our chalk rivers have such a huge base flow from groundwater, high contribution from groundwater, means that, you know, the mirroring of these trends are to be expected. And the third thing really just to point out is that seasonal variation we see in nitrate concentrations with time. So similar to the spot sampling that we were just hearing about, it's very important to look at those long trends. If you were just looking between 2000 and 2008, you might get a different picture of what's actually happening in the river. So just a quick chat about the hydrogeology and the recharge mechanisms in the chalk. So this is just a picture of the chalk at the coastline, but what's important is it's a Seaford chalk formation, which is the same chalk formation that we see in the Test and Itchen. This particular formation has lots of flint bands, and it also has quite a lot of vertical fractures and fissures compared to other chalk formations. So the recharge occurs predominantly through the matrix of the chalk. So as water infiltrates the surface, it moves through the matrix in a piston flow mechanism. So the water infiltrates the soil and pushes the water down through the matrix, and some water is released into the saturated zone. And we think the flow rates for this mechanism is about 1 meter per year. And obviously there's another pathway, a faster pathway for recharge to get to the groundwater, and that's through these vertical fractures and fissures, but this is very variable. But we think overall, on average, it might be about 15% of the total recharge comes down through these fractures and fissures. In karstic areas, that might be higher. So what does that mean for how chemicals move through our chalk aquifer? So in our unconfined chalk aquifer, nitrogen is in the form of nitrate, which is very soluble, highly mobile. So it basically moves down with our recharge. And so it moves through the matrix of the unsaturated chalk at approximately 1 meter per year. So if we have an unsaturated zone of about 50 meters, then that nitrate will take 50 years down, or approximately, to get down to the water, excuse me, down to the water table. We talked a little bit about seasonality earlier, and we do see this is a. Sorry, I should have said that. This is a graph, another one of our groundwater quality monitoring points in the Itchen. And we can see clearly here, again, the rising trend in nitrate, and the higher concentrations that we see in the wetter years, the lower ones in drought years. And this is basically when groundwater levels rise. The nitrate that's been working its way down through the unsaturated zone, moving towards the water table, is released into the saturated zone, and the nitrate concentrations rise. And I've also, just because it's a question I'm often asked about phosphate in groundwater, it doesn't react the same way. Phosphate is much less mobile, it's more reactive with the soils and minerals, in particular the chalk, where calcium phosphate is precipitated out and absorbed into the chalk matrix. So not a lot of phosphate generally gets down into the groundwater. And here we can see at this monitoring point a very stable trend, and quite low concentrations of phosphate. So onto the nitrate time bomb. We commissioned some WSP to do some nitrate modeling quite recently, just to show the impact of the nitrate time bomb, that legacy impact of agricultural practices, a few decades ago. I'm going to focus in again on Eastern. It's being picked on today in the Itchen. This is a map just showing, if you can see the groundwater catchment for Eastern here on the Itchen, and just to demonstrate, quite how large that groundwater catchment is, and the fact that it goes right up onto the high ground here towards Alton and Petersfield. This is a map of the superficial geology. So the brown is the clay with flints up there on the high ground. So a couple of points from the modelling that we commissioned is that firstly, we've had to assume that pretty much all the recharge and the nitrate travels through the matrix, because it's very tricky to model the nitrate pathways through the fissures and the fractures. It's very unpredictable. And the other thing is we could only have done this type of modeling that I'm about to show you because of that high groundwater contribution to the rivers in this area. So this is just the first graph I was just going to show you. So this, tricky to get your eye in, but the purple basically, well, it shows the age of waters arriving in the Itchen at Easton from the different land uses in the catchment. The purple is arable because that's the greatest land use in this catchment. And it shows that the age of water arriving at Easton can be really young, 5 years, to over 5% of the age of water arriving in the rivers as over 100 years. So obviously this younger water here will be originated in the lower lying land near to the river. And this really old water originates right up at the top of that catchment in that graph, the map that we were looking at, and has traveled possibly through 100 meters of unsaturated zone. And this graph shows, it's an attempt to show, what that onward trend might look like. This one assumes that there's nitrate leaching sort of continues roughly at the rate it is at the moment. And you can see that possibly trends start to stabilize, maybe decrease around 2060, 2075. And that slow decline is due to that combination of multiple age of water arriving in our rivers. So the range of ages in the river, it just demonstrates really that we need to be ensuring that nitrate leaching is reduced across the whole of our catchment. And finally, I was just going to talk about atrazine. So as most people know, it was a herbicide generally used to tackle weeds in crops like maize, and it was also used a lot in urban environments, especially on the railways and on the roads. And eventually it was banned completely in 2004. So here we have two graphs from our groundwater quality monitoring network again, one in the Itchen, one in the test. And you can see a downward trend there in atrazine. Now, obviously, the fate and transport of atrazine is a lot more complicated than nitrate. Atrazine is persistent and widespread. It's also the way it degrades to its degradation products or metabolites is very site-specific. And the one we see most commonly, the metabolite we see most commonly is desethyl atrazine or DEA. And we can see from this groundwater monitoring point in the test that the DEA is actually higher concentrations than the atrazine, but again, both on that downward path. And that was a very quick run through groundwater. Thank you for listening. And I'm going to hand over now to Sam. Thanks.
Great, thank you. 2 seconds. Right, hopefully you should be able to see my screen. Yep, I'm Sam. I'm a freshwater ecologist. I work for wild fish conservation, and I'm talking to you a little bit today about our smart rivers, system science monitoring, and chemicals in chalk streams. If I can get the clicker to work. There we go. Right, so if you don't know who Wild Fish are, we're an independent conservation charity working to protect wild fish and their waters. And within our work stream, Smart Rivers is our scientifically robust citizen science toolkit, for which we offer comprehensive training and support to try and empower local communities to collect professional grade monitoring data to take action to protect their rivers. And our approach generates data that matches the standards used by regulatory bodies like the EA, and we hope that we help to bridge critical gaps in monitoring caused by resource constraints. And so we're a national programme, we cover over 120 rivers, we've got over 30 amazing organisations that host our groups, and within that national programme we've got active monitoring on 18 chalk streams covering around 90 sites. We're based in Salisbury, so most of our these chalk stream monitoring sites are across Hampshire and Wiltshire, so the Avon, the Test, the Itchen and their tributaries, but we also have a couple of other groups scattered around. So we have the Great Stour in Kent, and we have the Wensum in Norfolk, and the Chess in the Chilterns, which we'll hear about next. So what Smart Rivers is invertebrate monitoring. So it entails going out in spring and autumn, collecting invertebrate samples using standardized methodologies, the kick sweep samples, and each of our samples is preserved to allow for the identification to be done to, where possible, to the species level, particularly for the river flies, which are the real canaries in the coal mine for pollution and invertebrate monitoring. So that's your mayflies, caddisflies and stoneflies. And what our monitoring produces is water quality scorecards. So these are metrics calculated from the invertebrate communities, covering things from siltation to chemicals, and then also kind of a view of biodiversity of the invertebrate communities, so profiles of diversity and abundance. And within the water quality scores, the main one relevant to today's talk is a metric called SPEAR, which is a species at risk, a system that was designed to assess the impact of pesticides on freshwater invertebrate communities, and it's a metric that uses biological trait information to identify these pressures and the effects in the rivers. And it's important to note that SPEAR can indicate a chemical impact at a site, but it won't tell you what the chemicals are. It just says there's a problem here. And across Smart Rivers, we've got lots of examples of highly impacted SPEAR scores in relation to known pollution hotspots. So that might be downstream of a dairy farm, for example, or sewage treatment works. And we've also found that although SPEAR is designed for pesticides, it seems to act as a surrogate for many other chemicals, potentially due to the way these complex chemicals interact with invertebrate nervous systems. And so here the graph on the right is showing at the top our SPEAR scores, and we present our water quality metrics in a kind of traffic light system. Green is good, red is bad, and the points of concern we have are the orange and red. So that's when what we consider a concerning score. So spring last year on the River Ailyn, sorry, Alison, this isn't the chalk stream, but there was a formaldehyde spill. Our volunteer group have gone out in late April and done their normal Smart Rivers survey. There was then the formaldehyde spill from a chemical company who initially said that none had got into the river, but turns out that was a porky pie, and lots got into the river, and our volunteers went out and were able to monitor again. And what you can see from these graphs is in the couple of weeks apart where the spill happened, you can see an increase in chemical pressure in May, and below we can see the river fly diversity and abundance. We can see a sharp drop in both of those biodiversity metrics. So chemicals in chalk streams, I'll skim over this because we've heard a bit already, but the main thing is it's an unseen threat, right? So the top image here on the River Till is this lovely clear chalk stream water that we love to see. You wouldn't be able to, from looking at it, know if there's a chemical issue in the water, whereas on the bottom, if there's kind of a build-up of growth on the substrate of the riverbed, you know that might be organic enrichment or some kind of nutrient imbalance. And on the right, if there's a sediment problem in the river, you can pick that up just with a visual scan. You can't do that with the chemicals. And as we've already heard from other speakers today, we were talking about things like the biocides, so the pesticides and herbicides we use that might be in agriculture, for example, in the chalk streams, particularly things like salad washing and watercress farms as well, but also in veterinary medicines. And while obviously livestock's a huge issue. We're a nation of pet lovers, and so the flea and worm treatments we put on our cats and dogs can end up getting into our rivers, be it from a dog taking a swim or indirectly through bedding being washed and entering the wastewater treatment system. And these are chemicals designed to kill invertebrates, so it's not hard to imagine what happens when they get into the natural world. And as has also been talked about, a huge array of human products, pharmaceuticals, personal care products, household products, mixing with forever chemicals and road runoff in the rivers. And then we start to get those cocktail effects that we heard about from Rob earlier on. And then, as we just heard from Polly, with the chalk streams, you also have this legacy aquifer contamination where banned compounds can end up appearing long after they're meant to have disappeared. And it's quite scary how many of these chemicals we see. I mean, we heard about Gary's data earlier, and we've seen ChemCatcher data at Wild Fish, that it's an astonishing number of chemicals floating around in the rivers. Why do we care? Well, obviously there's the kind of lethal effects of chemicals. Insecticides kill insects and other invertebrates, and so you can see biodiversity decline, and this will subsequently have knock-on effects on food chain dynamics and ecosystem services within the rivers. But something I find particularly concerning are the non-lethal effects that we get, and these it was touched on earlier, the endocrine disrupting compounds. So these are chemicals that interact with the hormonal system, and this is very much what makes the biological world go round. So we're talking about fundamental impacts to how organisms work. So this could be physiological impacts, cell growth, embryo development. It could also cause behaviors in animals that are not conducive to their survival. These chemicals will accumulate within the animals in the rivers, and these will bioaccumulate up the food chain and ultimately can even end up on our plates. And then another big issue, which we heard about earlier from Rob, is feminization, and we hear a lot about it in fish, but also happens in the invertebrates. So here, these are images provided by Dr. Cyril Bennett. We have a male large dark olive mayfly, which was found to be bearing eggs. So it can be easy to see what can happen at a population level when we have these strange quirks going on. So what impacts do we see in smart rivers? This graph is just showing all of the chalk stream monitoring that we've done since 2019. Each of these points is a survey. And as I said earlier, they're color coded from unimpacted or low impact up to bad. So green is good, red is bad. And what we're able to see here is that we're getting much more concerning scores for our spear metric being seen in our autumn surveys. So in 2021 was a particularly bad year, where about 70% of the sites that were impacted had a concerning level of impact seen. But since 2022, we're still sitting at about 60% of our chalk stream sites showing concerning levels of chemical impact in the autumn surveys. And it'll be really interesting to see what this year turns out like, because 2024 there was a lot of water in the rivers, a bit more dilution, and so over our monitoring network as a whole, we saw a reduction in impacts, and it might be a different case this autumn. And what we're looking for in this data is particularly sites that are repeat offenders, because that can really indicate that there's a persistent chemical issue going on in this area of the river. So how does this relate to the invertebrates? So what this is, these graphs are the same points you just saw in the graphs before. So each dot is a survey and a spear score for that survey based off the invertebrate community, green being good, red being bad. And here they're plotted against invertebrate biodiversity. So the two graphs on the left are diversity, the two graphs on the right are abundance. The top two are the entire invertebrate community, and the bottom two are the river flies, the canaries in the coal mine, for invertebrates as pollution indicators. And this is quite a coarse comparison, and the data needs tidying up, particularly in the abundance counts, because you can see we've got some quite large, quite a few quite large abundance counts, but you'll notice they're all green. and that's associated with an unimpacted SPEAR score. So what these graphs are showing are quite strong correlations where when you have a higher chemical impact according to SPEAR, you have reductions in diversity and abundance in the invertebrate community. And these correlations are stronger in the sensitive riverfly groups. But it's worth mentioning that obviously that the chemicals aren't the only pressure impacting the invertebrate communities for this data, but picking it out on the face of it, we do seem to see quite strong correlations with chemical impact. What else do we use the data for? So this is an example from the Great Stour in Kent. And so we can kind of take an average of chemical impact over seasons and years, and just kind of keep a bit of an eye on what's going on year to year, season to season. So for example, here in the spring surveys on the Great Stour, the last few years, there seems to be an increase on average in chemical impact, whereas in the autumn, there seems to be a decrease. So you can get different stories from the different seasons, and this might relate to work going on in the catchment in those periods of time of the year. And then the other thing that we really like to use the data for is looking at kind of a higher spatial resolution, so identifying particular sites of concern and thinking about what we might be able to do about it, what impacts it is having on the invertebrate communities. So this is the river Wensum in Norfolk from spring this year, and this is a similar graph to what you've seen earlier in the talk. Green is good, red is bad, and you can see on the top graph the chemical impact. At site 3, we've got the bright red bar, so this is showing a kind of poor chemical condition. And below we've got plots of diversity and abundance for the invertebrate communities across the sites. And you can see that site 3 has lower diversity and lower abundance compared to the other sites. And the colours in this, I don't concern myself too much, but this is within Smart Rivers, we train volunteers to identify 8 invertebrate groups, but within those groups is that really valuable species level data we collect, which lets us get kind of a true assessment of what's going on. Citizen science and chemicals, how can it help? So as I said at the start, SPEAR can highlight the chemical impact in an area of the river on the invertebrates, but it can't tell you what the chemicals are, which is obviously really important. So how these citizen science and vertical surveys can help is it can provide quite a cost-effective way of identifying areas of concern and chemical impact along a river to help target further action. And this data can be used for kind of local action, so engaging if we get repeat high chemical impact scores downstream of a particular input, we can use that data to talk to the people associated with that site and see what we might be able to do about it, but also use it as a tool for public awareness and education. And finally, the really strong point is that you can use this to prioritize your resources. So if you're able to conduct citizen science and vertebrate surveys that highlight a chemical impact on a certain area of the river, you can then use this data to prioritize your resources for more detailed and intensive investigation. So for example, Gary's chem catchers that we've heard about. If you're able to say that there's a repeat chemical impact at this area of the river, we should put our chem catchers there and see what's going on. Great, so thank you very much for listening, and I'll pass over to Kate.
Thank you very much, Sam. I will just share my slides. See if that works. Can you, no, you can't see those. Can you see the slides okay?
Yep, I can see that, Kate. It's just
Okay.
not in presenter view yet.
Is it in presenter view now? Not yet.
Not for me.
Now?
It is now, yep.
Brilliant. Okay. So I'm going to be talking about how local groups are understanding and addressing chemicals in chalk streams, and talking on behalf of the River Chess Smarter Water Catchments programme. So through funding from Thames Water for Chess Smarter Water Catchments, we've set up a citizen science programme on the River Chess that's been running now for over 4 years. And this is an integrated catchment management approach to improving the health of this chalk stream, which flows through the Chilterns National Landscape. And national landscapes were formerly areas of outstanding natural beauty. They've been rebranded as national landscapes. And the River Chess flows to the northwest of London. Now, Chess Smarter Water Catchments is a partnership approach. So this work today arises from the combined efforts of many different groups of people and organisations. So you can see some of the logos for the organisations here. And in terms of local rivers groups, we've got the River Chess Association, and we've got the Chilterns Chalk Streams project as well, who have been working in the catchment for quite a long time now on citizen science activities. So through Chess Smarter Water Catchments, we were able to enhance those citizen science activities and bring more things into play, which I'm going to describe a bit about today. And the Chess catchment itself is about 105 kilometres square. It flows for about 11 miles. It has its source in Chesham, in an urban area, then flows through sort of more rural parts of the catchment down to Rickmansworth and the River Colne. Now, the sewage treatment works is a red dot on the map there. It's a major influence on the river, comprising 40 to 80% of flow, depending on time of year and depending on rainfall in previous years. So I'd like you to keep that in mind as I'm talking about chemicals in the River Chess and the work that we've done. So when we started our citizen science programme, we first did a baseline assessment to understand what are the knowledge gaps on the River Chess. So what do we already know about chemicals in the River Chess and what don't we know? So we divided the river into reaches according to land use and critical river inputs and activities, and produced maps with monitoring activities already being carried out in each reach, for example, riverfly, and thought about what do we need in terms of frequency of monitoring and duration of monitoring on the river. We brought in data from the Environment Agency, from water companies, and from previous citizen science analysis as well to understand how we should be monitoring chemicals in the Chess and what were the unanswered research questions. So we based our citizen science activities around these research questions. So for us, it was really important to understand when do the storm tanks overflow from Chesham's sewage treatment works, for example. What's the effect of these storm tank overflows on dissolved oxygen levels in the River Chess? And are these storm tank overflows affecting invertebrate health? So we put together a programme of activity to try and investigate that further, along with some other questions as well. So for us, it was really important to install water quality sensors in the river so that we could understand changes in chemistry at high frequency. 15-minute data so that we could track storm events, for example, and understand what was happening when it rains, what is the influence on this chalk stream, which after all is groundwater fed. It's 95% base flow, but storm flows are really also very important in this river system. We had inputs from some of our citizen scientists, so some who are really expert in actual data analysis, for example, created us a dashboard to interpret the sensor data. But we also wanted to understand that link between chemical concentration and chemical patterns and invertebrate health. So we used and are using the Smart Rivers approach that Sam's just explained to you, and I'll show you some data from the River Chess using this particular toolkit. We put 6 sensors into the River Chess of different types, and these were monitoring and have been monitoring since 2019 chemical parameters like pH, dissolved oxygen, electrical conductivity, turbidity, ammonia, nitrate, tryptophan, which measures organic matter, and some derived parameters like oxygen demand, biological and chemical oxygen demand. And our citizen scientists have been trained in looking after these sensors and interpreting the data as well. We also have research scientists coming along every month to calibrate the sensors to ensure we have really good data quality and that that's sound. What have we used these types of sensors for? What kind of data are we getting? So we've been able to understand some of those reasons for the key chemical inputs to the River Chess. So for example, you can see a graph here that's showing some monitoring in December, looking at the blue line is change in temperature. So you can see that this monitoring took place during a time when temperatures went from close to 0 degrees centigrade to, and this is water temperature in the river, to 8 or 9 degrees centigrade. So essentially a period of snow melt. And you can see here that electrical conductivity spiked in this time in the river, showing road gritting salts entering the river system. So this is indicative of a route from road runoff into the river system, and showing how important it is to think about those chemicals that end up on our road systems. They make their way into the river via road runoff, and for example, during this time period as well, during snowmelt. We've also used the sensors to monitor storm tank overflow events in the Chess. This is some ammonium concentration data. So this is the green line here is ammonium. The blue line is pH. And you can see here two storm tank overflow events that happened in the River Chess in October 2023. So you can see there that there was one on the 20th and one on around the 29th of October. But you can also see that we have this big, broader dark green peak in that middle section as well. And this is just to show you how we've been able to work with the water company to get a quick response to events when they have happened in the River Chess. So here, one of the sewage treatment works dissolved oxygen probes had malfunctioned, created a false reading for the system, which believed there was enough dissolved oxygen aerating the treatment works, which there wasn't. So this resulted in elevated ammonia. We picked this up in our sensors and were able to have discussions with Thames Water to get that issue really rectified quite rapidly, and it was rectified on the 25th of October. So this kind of data enables us to have more meaningful conversations with the water company about what's happening in the river, and we have our own source of data to understand chemical impacts, for example. So that's the kind of thing that we're doing at the moment, and that's carrying on, and we've been doing that since 2019. Now, linked to that, as I said, we wanted to understand not just concentrations of chemicals, but also what the effects are on invertebrates in the river system. This is just a little conceptual diagram of the River Chess, and you can see there are two sites marked 2 and 3 on the river system there. These are sites where we take samples for smart rivers. So these samples are taken every 6 months. Site 2 is downstream of Chesham Sewage Treatment Works and receives water from the treated effluent from the storm from the sewage treatment works. Site 3 is on the Little Chess, which doesn't receive any effluent from Chesham Sewage Treatment Works. So just keep that in mind as I show you some of the next couple of graphs. So here we've got some of the results from Wildfish from the Smart Rivers programme. So it's great that Sam explained to you the colour coding, so I don't have to do so, but basically green is good, and then as you get increasing stress, you'll see sort of the graph columns coloured orange to red. So if you look along the bottom, you can see site numbers. So remember that I pointed out that site 2 is downstream of the sewage treatment works. And Wildfish methodology, the Smart Rivers methodology, gives you lots of different indices that you can help to interpret what's happening in your river system. So for example, we can see that there is stress in the River Chess arising from siltation. That's that first graph on the left. We've been able to track what's happening with phosphorus, for example. So you can see there that downstream of the sewage treatment works, there's the most stress in the river system from phosphorus. We're really interested in the Chess, where phosphorus stripping has just gone in, to understand how that index changes over time and whether it changes over time in response to phosphorus stripping at the sewage treatment works. And on the right, we've got the SPEAR index that Sam was taking you through. And again, you can see here that we have the greatest stress from chemicals immediately downstream of the sewage treatment works. And interestingly, number site 3, which is the Little Chess, has still got some indications of stress by chemicals. We've been able to use machine learning approaches to interrogate data from our sensors. So without going into this in too much detail, what's been interesting for us is to see how electrical conductivity reflects the activities of the sewage treatment works on the river. So the green and the purple lines here, you can see a kind of double peak every day from a change in electrical conductivity arising from treated effluent. So we tend to use, systems in the morning and in the evenings, and we can see a peak in solutes moving through the sewage treatment works as a result. And the blue and the red sites are above the sewage treatment works. So there's a kind of pulse in electrical conductivity, repeated pulse in the river every day. And we can use this to understand perhaps when to sample, for example, for those chemicals that are coming out of this system, to understand how best to sample to understand the effects on the river system. And we've also used this data to calculate how quickly water's moving down the river system. I'm not going to go into this slide a lot, but we're able to use machine learning to explain these water quality dynamics in this urbanized river and understand, for example, the effects of the wastewater treatment works on temperature in the river system. Now, we really wanted to understand what chemicals were causing the effects in the River Chess. So we had a lot of data from the Environment Agency relating to regulated chemicals, but we wanted to understand a little bit about emerging contaminants of concern. So we've worked with Imperial College and our citizen scientists went out for a year, taking samples every month from selected sites in the River Chess, and these were analysed for about 200 chemical compounds. Now, at the same time, we also used passive samplers, and we put those out twice in the river for a fortnight each as well, to capture more chemicals for presence and absence within the river system as well. Now, I think I have a slightly different view of spot sampling and passive sampling than I think Gary was talking about earlier on. I think when you have citizen scientists who are able to go out and take spot samples under different conditions within the river, you can actually make a lot of spot sampling. So we've done both approaches, and I'm just going to show you some of the results of our spot sampling on the River Chess. So what did we find when we aggregated all the results together? So remember, for each site, we've been out and our citizen scientists took samples, 12 samples during the year at a number of different sites. And we were then able to aggregate those samples to give a total average concentration of these 200 chemicals that we were looking for on the chess. Now, the graph shows you moving from the left to the right on the bottom axis, you're moving from the top of the river down to the bottom of the river. So from Chesham down to Rickmansworth and past Chesham sewage treatment works. We found a mix of pharmaceuticals, pesticides and illicit drugs as well in the River Chess. And you can see that the site labelled RHL had the highest total average concentrations of these chemicals, and that's immediately downstream of Chesham sewage treatment works. To the right of the site that's labelled RHL, you see a site that's labelled GWL. This is a groundwater spring. And by sampling this, we were able to get an idea of those chemicals that were in the groundwater of the River Chess. And then those sites to the right are moving further down the river system to Rickmansworth. And you can see there very little dilution by groundwater. These chemicals are maintaining their concentration as they move down through the river system. In terms of what did we find in terms of the chemicals, so in terms of low risk, now it's important to point out here, and as Rob said earlier on, you can find chemicals in rivers really wherever you look. We have very good techniques, very good analytical capabilities now to enable us to analyze chemicals at parts per trillion level in rainwater, in river water. And we need to think about the level of environmental risk from these types of chemicals. So Imperial College used a risk quotient methodology to say what risk these chemicals pose to aquatic life. And you notice there are no high risk chemicals on this list, which is great. So there were no chemicals thought of emerging contaminants thought to be of high risk to the ecology of the River Chess, but we did have some chemicals of low and medium risk. So atrazine, which Polly mentioned, was on there, a herbicide used predominantly to control weeds in crops and banned by the EU in 2004. We had various pharmaceuticals. These are of mid-range polarity, so they're not well removed by conventional sewage treatment works. So they were found, as you saw, downstream of Chesham. We, interestingly, for the medium risk category, we have a neonicotinoid insecticide, which is licensed for the use in the UK still and used to control sucking and chewing insects. And diclofenac, which is the active ingredient in products like Voltarol, found at relatively high concentrations, but only for 1 month in the river system. So we work with a citizen scientist to plot some of this data. Now you can see here 2 plots, one for the neonicotinoid insecticide that I mentioned, so acetamiprid on the left-hand side there. So that's a chemical that's in current use, and you can see that was detected in the river in autumn time in 2022. And in the river samples, but not in at low levels in the groundwater spring. Whereas if you look to the right-hand plot, that's for atrazine. That blue line is the concentration in the groundwater spring. Because atrazine is, as was explained to you before by Polly, is basically, this is a legacy chemical that's now in the groundwater and entering the river and potentially causing ecological impact in the river. So what did we do with some of this information? We now know a lot about emerging contaminants within the River Chess, and we can link that also to how these chemicals are getting into the river system through the other work that we've done with the water quality sensors, and understand a bit about the effects of these types of chemicals on the river through using Wildfish Smart Rivers approach. Our citizen scientists decided that they would really like to produce some leaflets on what we can do for the river. So we've got leaflets telling about what chemicals we can use in homes and gardens, and what we should be thinking about. So for example, not flushing unused medication. As I think Rob mentioned at the very beginning of the talk, considering non-topical flea treatment. Our passive samplers showed presence of flea treatment chemicals, and limit use of bug sprays and consider alternative garden sprays to those that contain acetamiprid, for example. And finally, we've published all of our findings in a report on citizen science on the River Chess, and you can visit our website to download a copy of our report if you're interested in what we've found. So I'm going to hand over to Rob now.
Thanks very much, Kate. Perhaps if all the presenters could put their cameras on, I think that would be helpful. We've got about 10 minutes or so to just run through some of the questions that have been flagged in the Q&A. And just to say, there are some quite technical questions specific to particular presenters. So if you could just have a quick look through some of those. But one I picked up on, a couple of people raised this around pet parasiticides, flea and tick treatments. And I have put a link on there. The government have established a roadmap over the summer to begin to look into these treatments. But there's a question there around oral treatment versus spot on and so on. And I know Isla Thorpe, as a student at York University, has been looking at this and looking at wastewater discharge of pet parasiticides versus sort of dogs in rivers and so on. But I just wondered if any of the panel wanted to comment on this. And I'd flag also that I noticed the government document says they're looking into trying to establish a PNEC, predicted no effect concentration, for these chemicals. So perhaps Gary, you might want to comment on that last point, but others might want to jump in.
Yeah, I can jump in. So one of the government's looking at, well, certainly for imidacloprid, they're looking at it in terms of a neonic and impact on bees and pollinators, but there's still, I can't find anywhere in the literature or government stuff that says anything about potential impact on aquatic ecosystems. They're still coming at it from a bee and pollinator one. I mean, there is there are PNECs for imidacloprid out there, certainly in the database we use for Norman, but whether there'll be one that will actually be something in Water Framework Directive, whatever the UK version of that is going to be, I don't know. I mean, somebody else asked a question about the kind of the impact in are there studies. So there's lots of studies on imidacloprid having and fipronil having impacts on aquatic organisms. So that's one of the, I mean, there are, in terms of the various impacts on organisms in the aquatic ecosystem. There's lots of work being done at Imperial as well, as you mentioned, so as well as the group at York and Imperial. And so, I mean, it would be, I mean, ultimately, I think there needs to be some ban or potential ban on the use of the spot-on treatments. I mean, we've worked with Leon and we've seen Leon give his presentations about that. But it seems that there's lots of people use those treatments that still, aren't aware of the impact that that's potentially having. I mean, imidacloprid was banned 2008 or something in terms of agricultural use, but I think, oh, Sam.
I was just going to jump in after you. I think you're spot on. You can buy imidacloprid off Amazon with no kind of checks, but you can use it in agriculture. I think the kind of the questions are also about kind of oral use versus spot on, and I'd say that if you're thinking about, say, your dog going swimming in a river, an oral use is going to have a less direct impact than a spot on washing off into the river. But Dave Golson's research group from Sussex has shown that one of the main pathways is it coming in through pet bedding. And as with all drugs and anything we take, anything our dogs take, even if it's oral, there will be waste products excreted through sweat or waste products from your pets. So they'll still be able to enter the system that way. I think one of the ways with pet protection products is us using them sensibly as well. So, rather than monthly subscriptions at your vets, just treating flea and worm constantly, that we maybe treat our pets. If our pet has fleas, we get flea treatment. I live up in the borders of Scotland, and I treat my dog for tick medicine orally. I only do it in the kind of peak periods of the year where the risk is highest. I'm not doing it 12 months a year just in case, because that's where we're getting these kind of excessive amounts entering the systems indirectly.
Yeah, thanks, Sam. A couple of questions about water company data. So what do they do, and are we involved in chemicals investigation program? And the chemicals investigation program, I guess, is the most freely available, transparent data that chemicals data we can get access to, is one thing. In terms of how closely involved with that, we in the Rivers Trust, working for the Wildlife and Countryside Link, we've been involved in discussions with Water UK about advocacy of the government around tackling chemicals at source, particularly PFAS, but others. But I wondered if others on the panel other presenters have got any experience here or thoughts around water company data, and what else might we want them to do, or what are the gaps? We talked about pesticides and linking pesticides to raw drinking water sources and so on, for example, but any thoughts on that one?
Yeah, I can jump in on this one. So we've got that project on the Hampshire Avon, which is always confusing. So I'll call it the Hampshire Avon, but we're sampling in Wiltshire, but it's the So we're working around that Amesbury area, but we're working with Wessex Water, and so some of the stuff that we're doing is kind of informing them and working with them on the investigation program in terms of what contaminants, how we can link that data together, what's coming out of the effluent pipe. Are we looking at the correct ones in terms of doing some screening, which depending on what they're actually targeted to look for in that chemicals investigation program, particularly in terms of the emerging contaminants. So are we looking at the correct ones? And then linking that to, I mean, ultimately, is there any way that you mentioned, Rob, is there any way of tackling that at source? What kind of treatment do we need to have in the water recycling centre, as they like to call it now, rather than the sewage treatment works? What kind of technology do we need to be able to maybe tackle that? So it's going to be difficult to stop people from using things, but if we can, I mean, it's going to be expensive, but if they can up their game in terms of processing at the water recycling centres, can we then start to take these important contaminants out before they then come out of the effluent pipe?
Yeah, definitely. Thanks, Gary. I'm going to ask Kate a quick question. It's somewhat related to one of the Q&A questions, but your sort of public-facing program, Kate, awareness raising. I really like the leaflet and so on you created. Have you? It's probably a tricky question, maybe an unfair one. Do you know how well you managed to reach out into the community? I guess it's a big job, and how well you can change people's behavior by this. It feels very important to me.
Yeah, and I think we've only just started, Rob, with producing the leaflets. The leaflets for us at the moment have gone out to local libraries, to local community groups, but we haven't done that big push in terms of trying to get more information out there. I think for us, it's really important to understand how the results on the chest link to other people's findings. For example, for Isitamiprid, what exactly is happening there nationally, and then to push a campaign more nationally as well. I think it's always really useful. I think some of the things that we're seeing on the chest are definitely echoed in other chalk streams around the country. So it would be great to work on how do we encourage people to change their behaviours around their homes with these chemicals as well? Because as you said earlier on, we've got the regulation side, that's absolutely critically important, but we also need to think about what we do, because I think it's going to be incredibly expensive for water companies to tweak tertiary treatment to remove all of these types of mid-polarity chemicals that move through the works. So there is a kind of need to do both things. So yeah, in essence, no, I don't know how well our campaign, has been successful so far. It's just the beginning of sort of raising awareness about these things. But I think it was great that the citizen scientists have taken forward this idea of what do we do with the results? How do we make a change? Recognise the purpose of the regulation, but also that there is something in how we choose to use chemicals around the home, and informing people and helping people make better informed decisions.
Yeah, thanks, Kate. Yeah, really, really crucial stuff. Perhaps one real last quick question. What's the future in terms of citizen science and chemicals monitoring? We've had chem catchers, passive samplers, the work you described, Kate. Are we moving towards a lab on a chip type thing? Can we see it envisage? I mean, I think Ruth, it was, who asked a question about, how do you go about doing this sort of stuff if you haven't got chem catchers to hand and whatever? Are we seeing a direction of travel where it's increasingly going to be up to non-regulators to, get this data out there?
So we certainly had to work with the universities to do this type of work. I mean, you've got to be very careful how you collect the samples, right? These chemicals are at very low concentrations. You need to be trained up well to make sure that you don't accidentally contaminate samples as you collect them. So there's that aspect. Sample volumes are getting smaller and smaller, which is great. So citizen scientists can go and collect very tiny volume water samples, don't need to collect sort of litres of water as they did in the past. But I think at the moment, there's a limited number of chemicals through chemical on a chip, on a lab on a chip type analyses, and we're still kind of working where we're either using passive samplers to absorb chemicals and taking those back to a lab for analysis, or small water samples. Gary may have an idea of newer innovative technologies that are coming along, but certainly those are the two things that we've looked at and used.
Oh, I think that those are the two main ones. I mean, there's some development work for some certain contaminants of emerging concern, but virtually all of the ones that we're interested in do need some kind of big chromatography instruments. So I would say, although they do, for the Environment Agency have now got a number of new analytical facilities, so they will start doing more screening. So as well as just the way that you sample it, it's also the way that you analyze it. So if we can now do more suspect screening, non-target screening, there's going to be a lot more data out there, and that's what the EA are now starting to get online now, is improving the analytical capability which they now have, to try and we refer to it as more of the chemical fingerprint, because people were asking questions like what chemical is impacting certain species, and it could be that it's something that we're not even analyzing for. So case in point is that 6-ppd, which there was impacting in I think the Pacific salmon, that nobody was sampling for. No one knew what it was, and then they discovered that it was that one. So we're probably looking at there could be chemicals that we're missing that we've never studied before. It could be the one, just the whole accumulation of all those different chemicals having an impact on the ecosystem as well.
Yeah. Okay. Thank you very much, Gary, and to all the presenters. And I'm conscious I need to hand back to Alison. Plenty of more questions in the Q&A, lots of specific ones, but we'll have to leave it there.
Yeah, thanks, Rob. And a huge thank you to all the presenters. I think it's really interesting to see that coming together of everybody across the academic, regulator, ENGO and community groups is really the way that we're going to solve this, and the water companies too. And so I think, yes, there's still quite a lot of open questions. I will take them away. I think there's something here about building a bit of a community and sharing information and knowledge, which is really important. And that's come through from the questions. We shouldn't all be trying to tackle it all on our own. So I'll try and have a think about how in the Cabba Chalk Streams group we can support all of you with that in whatever way we can. And I think somebody said this is very depressing. I think it's easy to feel a bit like that. It is. So I think the hope is that by working together, we can understand it better, and we can start trying to put things in place to try and address some of these problems, and hopefully give a bit of cause for optimism maybe in the future. But as Polly said in her presentation, these things take a long time to turn around. So it's not going to be a quick fix, but by being aware of it, taking action and doing something, I think we can all start to feel a bit more positive. So a huge thanks for joining us this morning. It's been really, really interesting. I've thoroughly enjoyed it. Thanks to all the presenters. And if people have got feedback or comments they want to make, then stick them in the chat, or do get in touch. And of course, the recording will be available afterwards with the presentations for those who want to share. So thank you very much. Enjoy the rest of your day.